This is a mentor and serve production. How the acid or H+ secretion occurs from the stomach and the mechanism of it. Hi, friends, let us see how this secretion mechanism works in the gastric tract. Parietal cells in the stomach secrete roughly 2 liters of acid a day in the form of hydrochloric acid. The acid in the stomach functions to kill bacteria, and to aid digestion by solubilizing food. The acid is also important to establish the optimal pH, between 1.8–3.5, for the function of the digestive enzyme pepsin. A key protein for acid secretion is proton pump. This protein, which is expressed on the apical membrane of parietal cells, uses the energy derived from ATP hydrolysis to pump hydrogen ions into the lumen in exchange for potassium ions. Stimulation of acid secretion. It involves the translocation of proton pump to the apical membrane of the parietal cell. When the cell is resting, not stimulated, proton pump are located in vesicles inside the cell. When the cell is stimulated, these vesicles fuse with the plasma membrane, thereby increasing the surface area of the plasma membrane and the number of proton pumps in the membrane. There are 3 regulatory molecules that stimulate acid secretion: acetylcholine, histamine, gastrin, and 1 regulatory molecule that inhibits acid secretion, that is somatostatin. Acetylcholine is a neurotransmitter that is released by enteric neurons. Histamine is paracrine that is released from ECL, enterochromaffin-like, cells. Gastrin is a hormone that is released by G cells, endocrine cells that are located in the gastric epithelium. Somatostatin is also secreted by endocrine cells of the gastric epithelium. It can act as either a paracrine or a hormone. Stimulation of acid secretion can be studied in 3 phases. 1. Cephalic phase. Cephalic phase stimuli are things like the sight, smell, taste or thought of food. These stimuli, processed by the brain, activate enteric neurons via parasympathetic preganglionic neurons traveling in the vagus nerve. 2. Gastric phase. The primary factor during the gastric phase is that there is food in the stomach, which stimulates acid secretion. There are 3 different ways that this occurs. Food will stretch the walls of the stomach. This is sensed by mechanoreceptors, activating a neural reflex to stimulate acid secretion. Purple. Peptides and amino acids in food stimulate G cells to release gastrin. Food also acts as a buffer, raising the pH and thus removing the stimulus for somatostatin secretion. 3. Intestinal phase. Once chyme enters the duodenum, intestinal phase stimuli activate negative feedback mechanisms to reduce acid secretion and prevent the chyme from becoming too acidic. This occurs by neural reflexes and hormonal reflexes. Enterogastrins are hormones that inhibit stomach processes, in this case, acid secretion. In addition to their other actions, CCK, secretin, GLP-1, and GIP act as enterogastrins.